So we start off at the very, very top. You see the DNA helix. This DNA contains information about how to make different protein structures. It's going to tell the body exactly which amino acids we need and which order to put them in so that the structure can be formed properly. So the first thing that we're going to do is we're going to go ahead and make a copy of this genetic information. And that copy is called mRNA. So we are going to make a copy of DNA. That copy is called mRNA. And this process of making the copy we call transcription. So that's step one. Step one is transcription, which is making a copy of DNA. And that copy is called mRNA or messenger RNA. The reason it is called messenger RNA is because it's actually going to take this information over to our protein-making factories, which are the ribosomes. The ribosomes are where we build our proteins, and so they're the ones who need the information on how to build the protein structure. So the messenger or mRNA is going to relay the information to the ribosome. And that is our step 2 that we call translation. Now, after that, the ribosomes now know exactly which amino acids they need and which order they need to put them in. So they're going to go ahead and start calling out on these amino acids. Now, we have some other helpers that we call tRNA or transfer RNA that are going to be waiting around to help out in this process. These are the little green things that you see hanging around with different colors on top of them. Now, the different colors signify different amino acids, and what they're doing is they're waiting around until they can deposit the amino acid that they are responsible for. Each tRNA is responsible for a specific amino acid. For example, the one that is carrying the red amino acid will always only carry a red one. So it's going to wait around until the ribosomes say they need a red amino acid, then it's going to come in and go ahead and drop it off. Then it'll go over to the amino acid pool and pick up another red one so that the next time ribosomes call for red, it's ready to go. And that's what's going to keep happening. The ribosomes call on the different amino acids, transfer RNA goes over to pick them up and put them in place. And we're going to keep doing this until we have a string of amino acids long enough and complete to build our protein structure. Now, sometimes there is an error in this process, and that's when we can develop things such as sickle cell anemia, which you see in this picture over here. Now, on the right-hand side, you see what a normal red blood cell should look like. And on the left-hand side, you see where there was an error in the making of this cell, and it's a lot different structure than what it should look like. The reason this happens is because the people who develop this have some kind of genetic defect where the DNA actually is wrong when it tells the mRNA how to make the protein structure. This is what we call a sequencing error. It's when the DNA will say, for example, put the red amino acid before blue when it's really supposed to be blue before red. So the amino acid sequence is wrong somehow. And because of this, remember, all the processes that happen after that are based off of the directions of DNA. So if DNA has something wrong, the way that we build the protein is going to be wrong. And we can result in a protein structure that is not what it's supposed to be. And that's what we see here. Because there was a genetic error in the DNA that told our body to make the red blood cell using a different sequence of amino acids, the red blood cell actually came out looking a lot different. Now here you see why this is so important, because at the top you have the red blood cells that are normally developed that can flow really freely through the blood vessels. But the ones on the bottom since they have this odd shape, they can actually get stuck. They can lodge themselves in the different areas of the blood vessels and actually get stuck and block blood flow.